Hello students, today we'll discuss about RNA processing. As if we have earlier studied about transcription, that how the RNA is formed from DNA. When RNA is formed, then there are different kinds of RNA which are formed. Sometimes it is like we can see mRNA, sometimes it is tRNA, sometimes it is like we can see it is rRNA. Actually, rRNA, tRNA, mRNA, and there are a number of other kinds of RNAs which are formed after the transcription. But the RNA which actually takes part directly or in case of translation is that is mRNA. But the mRNA which is formed after transcription, that is not the actual form of the mRNA, or we can say that is not the mature form of the mRNA, which is going to take part in translation or for the protein synthesis. So we should know that what actually RNA processing is. The pre-mRNA, which have been formed after transcription, that need to be converted to its mature or functional form to take part in translation or protein synthesis. That process is known as processing. So, first of all, we should know that what kind of steps or what kind of events occur during mRNA processing. But before moving towards this, we should know that mostly newly synthesized RNA must be modified in various forms to be converted to their functional forms. But when we talk about the bacterial mRNA or about the most of the prokaryotic mRNAs, they act as exceptions because they do not get converted into mature RNA, the kind of mature RNAs, or they do not undergo the process of RNA processing. They directly take part in translation just after transcription. But when we talk about the eukaryotic mRNA, they basically, either it is eukaryotic mRNA, tRNA or RNA, they undergo a series of steps to get themselves converted into mature or functional forms of mRNA. So when we are talking about this kind of events, they basically include the processing of the mRNA. The first event during mRNA processing is 5' capping. During 5' capping, actually 7-methylguanosine is added to the 5' end of the mRNA. When we talk about the second event of mRNA processing, which is polyadenylation, that is actually the addition of poly A tail to the mRNA. And last but the most important step of mRNA processing is splicing, in which introns are removed and only the exons are capped after this. When we are going to talk about RNA processing in detail, we should know that the mRNA formed directly after the transcription, that is known as pre-mRNA. Why it is known as pre-mRNA? Because it is not the actual mature form of the mRNA. So this mRNA, pre-mRNA, consists of exons, this consists of introns, and it is not having any poly A tailing, and there are a number of other reasons. First thing is, when it is going to be converted into mature RNA, first of all, it will undergo the process of 5' capping. As if you can check at the 5' end of the mRNA, there is addition of 7-methylguanosine, and a poly A tail is added at the 3' end, and after these steps the introns are removed and ultimately it leads to the formation of an mRNA which is lacking introns and which is having exons only. And after the conversion of the pre-mRNA into the mature or the functional form of the mRNA, it is transported from the nucleus to the cytoplasm of the cell, because ribosomes does not exist inside the nucleus, they are present outside the nucleus. And actually, these ribosomes are the main organelles which play a role in the synthesis of the protein, or in other words, we can say which act as the site of protein synthesis or translation. So mRNA, after its maturation, needs to be transported from the nucleus to the cytoplasm. This 5' capping do. This 5' capping stabilizes the RNA, and the second important thing is that it aligns the eukaryotic mRNAs, or it helps in the alignment of eukaryotic mRNAs on ribosomes during the process of translation or during the process of synthesis. Now, the second important event of what our RNA processing is polyadenylation. As if the term explains itself, polyadenylation. Poly means a number of. Adenylation means a number of adenyl groups or adenines will be added to the mRNA. After what? After the 5' capping of the mRNA. So, we are having this we can say pre-mRNA, and in this pre-mRNA what will happen, we'll see that the 3' end of most eukaryotic mRNAs is defined not only by termination of transcription but by cleavage of the primary transcript and addition of poly A tail, which is known as polyadenylation. But as if we can check from this flowchart that our pre-mRNA consists of some highly conserved hexanucleotide sequences which act as signals for polyadenylation, and these sequences are AAUAA. You can check this part as highlighted with yellow color in this flowchart. And this signal for polyadenylation, or this sequence which acts as signal for polyadenylation, is located approximately 10 to 30 nucleotides upstream of the site of polyadenylation. And in addition to this, a GU-rich downstream sequence element is also present over there. So what actually happens is, in this process, that first this primary transcript is cleaved, and this primary transcript is cleaved with the help of an enzyme known as endonuclease. And then the role of the enzyme that is known as poly A polymerase occurs, and this poly A polymerase is going to add a poly A tail to this mRNA. And now we can see that the first two events of RNA processing have occurred. First one was 5' capping, and second one is polyadenylation. What basically polyadenylation do? It regulates translation, and second thing is it basically stabilizes the mRNA, and last one is early development. When we talk about these three events, what actually happens is that when we talk about that this mRNA, what it is going to do, or what this polyadenylation is going to do. For example, we'll discuss one example, that many mRNAs are stored in unfertilized eggs in an untranslated form. And when we talk about these untranslated forms, they are with short poly A tails. They are having poly A tails of approximately 30 to 50 nucleotides long. When fertilization occurs, This fertilization stimulates the lengthening of poly A tail. Fertilization stimulates the lengthening of poly A tail of the stored mRNAs. And this polyetailing or this process of polyadenylation, it activates the translation of these mRNAs, or we can say these untranslated mRNAs which were stored in the unfertilized eggs. And the synthesis of proteins which are required for the early development of the embryo actually starts after the polyadenylation only. It cannot start itself. And the last but not the least, this event which is known as splicing is the most important part of the RNA processing. When we talk about splicing, first of all, we should know that actually after the transcription, the RNA which we are getting is that consists of two kinds of sequences. One are the coding sequences and another one are the non-coding sequences. When we talk about the coding sequences, they are the exons, and the other one are the non-coding sequences. And these non-coding sequences, we can consider them sometimes as junk sequences because they don't have any role in the formation of proteins or in the synthesis of proteins. They are not going to take part in the process of translation. So during splicing, what happens in case of eukaryotes? These non-coding sequences or the introns are removed. And this removal of the introns leads to the actual functional form of mRNA, which is known as mature RNA. So ultimately, we can see that here the mature RNA is differing from the primary transcript of the mRNA in three factors. First, this mature RNA is having what? 5' capping, and that is 7-methylguanosine at its 5' end, and it is having a poly A tail at its 3' end. And the most important thing, it is not having any kind of introns in it, or in other words, it is having only the coding sequences and no non-coding sequences present in this mature RNA. So ultimately, the protein formed will be consisting of or will be complementary to these coding sequences. When we will talk about this, like we can say we are going to talk about splicing in detail, we should know that splicing actually takes place in two steps. The first step of splicing is that pre-mRNA is cleaved at the 5' splice site, and here in this figure 5' SS is marked near the exon 1, that is the actually 5' splice site, and there is one more 3' SS which is near the exon 2, that is known as 3' splice site. Now we'll say that in the first step pre-mRNA is cleaved at the 5' splice site, and the 5' end of the intron is joined to an adenine nucleotide within the intron. Next step is that after this, what is going to happen is that the cleavage of the simultaneous cleavage of the 3' splice site and ligation of the two exons is going to occur. If we keep a check on this diagrammatic representation carefully, what we'll see, first of all, the cleavage will occur at the 5' splice site, and this end of the intron, that is 3' end of the intron, is going to bind with the exon 2 or nearby exon 2. But how will it bind with some adenine present in this intron? It will cleave from this exon 1 and it will bind somewhere in the intron itself, where an adenine is present, and that point is known as branch point. And you can easily see that ultimately it is leading to the formation of a structure which looks like a lariat structure. So now we can see we are having exon 1 separate, but the intron is still attached with the exon 2. We need to break this bonding between the intron and the exon 2. So how it can be broken? It can be broken with the help of some particular proteins. We'll discuss in detail it later on. So what will happen? This intron and exon 2 will also cleave from each other, and ultimately both of the exons will ligate to each other, and this lariat structure of the intron is ultimately. Now we'll move towards that how actually the whole process of the splicing takes place. Actually, there are a number of proteins which are going to take out or carry out the process of splicing. They need the help of a number of proteins, and these proteins ultimately they form a complex which is known as spliceosome. And these spliceosomes, they consist of actually some proteins, approximately 10 proteins, and some small nuclear RNAs. And these small nuclear RNAs are actually 5 in number. So we'll start step by step. What will actually happen? First of all, the U1 small nuclear protein will come and it will bind with the ligation point between the exon 1 and the intron. Or we can say the U1 small nuclear protein ribonuclear protein molecules, or in other words we can say U1 snRNPs RNAs is going to bind with the 5' splice site, and then there will be addition of U2, and this U2 snRNA is going to bind with the adenine present at the branch point of this primary transcript of mRNA. So what will happen? These U1 and U2 snRNAs will interact with each other. When they'll try to interact with each other, this will lead to the bending of this primary transcript, and it will form a hairpin loop-like structure. Then what will happen? A complex of a few snRNAs, that is U4, U6 and U5, they'll come and bind with this U1 and U2. What will happen? The binding of U4, U6 and U5 will lead to the release of U1 and U4 snRNA, and this will lead to the formation of a lariat structure, lariat-like structure, and the cleavage of the introns from the exons, and ultimately